So again, this just gets a little bit deeper. Care pathway development, and I touched on that in the previous slide. What happened, it's not just developing the test, it's the service around that test, right? Being the champion of optimizing processes up front, optimizing reporting processes and care pathways on the back end with the information you're generating through biomarker testing, right? It's strategically integrating genomic technology with the people and the processes beyond the lab, both up front and on the back end, to realize that promise of precision medicine for each unique patient. And so some of these pre-analytic considerations, certainly specimen acquisition is one. It happens differently in different places, right? I'm in Vermont, down the road from me where I did my residency at Dartmouth. We have very different practices around how we use cytology at our organizations. We use different staining types. It's almost like two different worlds, even though we're both considered, we're neighbors. So I think specimen acquisition is really important to understand. Specimen types are really important to understand. The way I validated our genomic assay takes into account what cytology stains we use here. They're very different than what Dartmouth uses down the road. So it's a good example, right? And then what testing portfolio is wanted? Sometimes if you're at a smaller organization, your clinicians may want just what's in the NCCN guidelines. Don't give me anything else. I don't want to risk, anything else. I don't know what to do with anything else. Or if you're at a place like, let's say, Memorial Sloan Kettering, right? Like where they have a massive portfolio of clinical trials, their clinicians may want a lot more than what the clinicians in Vermont may want. So I think working through what are in the guidelines, what the clinicians want to get, and coming to an agreement around that pre-analytically, because that will determine really then how much tissue you're going to need to go into the assay and be successful. So then if you have a bunch of those pre-analytic considerations, who's going to touch that tissue when it comes into the lab? I'll tell you, typically anatomic pathology will, certainly surgical, sometimes cytology, a lot of times cytology. Sometimes a specimen is shared even with laboratory medicine, right? They get a little aliquot of something. Certainly molecular diagnostics, like we're going to want a little bit of that. Sometimes there's a send out test. That's something special that's added on, and that has to go out as well. And then research and biobanking, depending on your local protocols. What are you doing with that? So there's a lot of competing interests in that very small biopsy. So if you're successful in sort of managing that and having awareness and transparency amongst these groups, and again, sometimes people consider this, oh, it's just the lab. Well, it's way more complex than that, right? And if you're successful in doing that, and you can generate a advanced biomarker profile inclusive of PD-L1, right, for non-small cell lung cancer, what are the post-analytic considerations? Generating a report, how do you do that? How is that report? How do you ensure it's visible and usable at the time when it needs to be? And also, I think data searchability, and not just for research, right? You may say, well, we're not a research organization, or that's not our primary thing, we're not academic. But I think every single person on this call understands you have to get at your data to do continuous quality improvement. So set yourself up for success in your system so you can access that data without doing what we call data dumpster diving. And then, certainly I think what's usually a hindsight consideration, and I think we need to be more thinking about this up front, is business intelligence. We should have an ongoing relationship in terms of molecular pathology, since we're on this like cutting edge, right? With our leadership to say, okay, you've invested in us to do this, right? We're requesting resources, you've given them to us, we've done something with it. And then to be able to use your data and turn around and work with business finance people to show, well, what's the value here, right? Some business intelligence. And underneath all of this, right, this can't be done in a sustainable way and a transparent way without having and leveraging our protocols being built properly and implemented into information systems, whether it's the lab information system right, the electronic health record as well. And all of this takes really this sustained education. Why are we doing this again, right? People in AP or perhaps, the send out groups or the biobankers, they're not always thinking first and foremost about, the practice of molecular and how you get these biomarkers done. So it takes consistent education.